Our research presenter today is physician scientist Dr. Andrea Amalfitano. Andy earned his Bachelor of Science and PhD degrees in microbiology from Michigan State University, and he earned his Doctor of Osteopathic Medicine, or DO, medical degree from the MSU College of Osteopathic Medicine's DO PhD dual degree program. His clinical and research training included a pediatrics residency at the Mayo Clinic and a clinical genetics fellowship at the University of Michigan. Prior to joining MSU as a faculty member, he served as an associate professor at Duke University Medical Center in the Department of Pediatrics and the Department of Pathology, Molecular Genetics, and Microbiology. In addition to his clinical expertise in seeing patients affected by a variety of genetic diseases, his research focuses on determining the feasibility of cutting-edge therapeutics, including gene transfer, to treat both acquired and genetic diseases, and then translating that knowledge into the clinical realm. His scholarly efforts in both clinical medicine and applied genetics have led not only to hundreds of peer-reviewed manuscripts, abstracts, presentations, and book chapters, but also a number of patents and numerous grants, including those from the NIH and private foundations. Together, these efforts have led to the development of innovative insights as well as treatments for infants, children, and adults affected by a variety of diseases, including lethal muscular dystrophies, cancer, autoimmune diseases, and autism. Please welcome Dr. Amalfitano.
First, thank you, Dr. Shu, for the introduction. Thank you, board, for taking some time out to hear one person's experience in translational research. And just to define that, I think translational research reflects the land-grant mission of Michigan State. That is, how do we take knowledge and move it to help the citizens of the state of Michigan, the nation, and beyond, and improve their lives. That's something that's been ingrained in me since my undergraduate days here at Michigan State, and I've always wanted to pursue that. And so I'll give you 3 examples of how this has played out in my career, and hopefully we can replicate this going forward. So I am a physician. I see patients in my clinic. I have the great privilege of bringing knowledge to them to help them even in times when a genetic disorder is diagnosed and we have nothing but supportive care. I can bring knowledge to families. That is fulfilling in and of itself. As a scientist, I want to generate new knowledge and tackle those problems that I encounter every day in my clinics as well that still have no solutions. And so I'll give you 3 examples of how this has played out. I've participated in clinical trials research for new drugs. I've invented new technologies, as we've heard, and also taken advantage of clinical patient data and noticed problems or questions that are there and used my background as a scientist to bring that to the laboratory, investigate it, and then maybe move forward with new understanding. So the first thing I'm going to talk about is a disease called Pompe disease. This is a brutal genetic disorder that affects families. This child, unfortunately, has the condition. What we're showing you here is a child that is very weak, can't control their head movement when you pull them up gently, has a feeding tube because it can't swallow correctly. Eventually, this muscular dystrophy, it involves their muscles, involves their heart muscles to the point where their heart can't function correctly, or their lungs are so affected by respiratory weakness that they, unfortunately, this is a lethal condition. At least it was. At the time, when there was a theory that maybe if we could develop a new enzyme therapy, a protein, if you will, the theory was if we could inject this into patients that have Pompe disease, this is the enzyme that they're missing due to their genetic background, would this drug pass into the bloodstream and go to all those muscle cells and reverse the effects of the disease? That is, give them back their genetic information that they're missing. And so I led the first in-human trial of just such a drug. And here's an example of how it worked in one of our first patients. On my left, on your left, if you're looking forward, you can see the heart of one of these patients is very large, obstructing the lung. And on the right side, you can see how much dramatically smaller the heart is. It should be fairly obvious. In fact, this occurred over 12 weeks. So we infused this medication twice a week to this infant, and within 12 weeks, we saw a dramatic improvement. In fact, this child is about 18 years old now, and continues to receive this treatment. It's just dramatic. In fact, we've sent him into pediatricians' offices and not told them the diagnosis, and he comes out with a clean bill of health. So based upon this first study, we were able to then move on to expanded studies, greater numbers of patients, and in fact, international studies to finally get this drug approved by the FDA and the European Union. This is the technical term, L-glucosidase alpha. We love coming up with these crazy names for these medications. But this is now a drug that's approved for all patients in the world with Pompe disease. And so physicians now can prescribe this across the world. In fact, I had a patient yesterday that I saw that I prescribed this to. So it's pretty exciting to be able to not only be involved at the initial stages, but now in my everyday clinical practice, I can actually bring this drug to patients in the state of Michigan, and I know that there's hundreds of other physicians doing the same thing. In fact, as another level of translation, we want to move this from clinical practice to general sort of practice nationally. In the state of Michigan right now, I sit on the state newborn screening panel, which decides which diseases are screened for in babies. And now I can tell you, every baby in the state of Michigan, within 24 hours of being born, is identified. And if they have Pompe disease and if they're at risk, they can get this treatment within days of birth. So we know the earlier we get in there, the better we intervene, the better the outcome. And so that is an example of how something can move from basic research, experimental clinical research, all the way to health policy and changes. I'll give you another example here of what I like to mess around with in the lab. Back in my microbiology days, you'd learn about how many people have had a cold recently, right? Many of you have. Well, what that cold is caused by is a virus, a cold virus that is on the outside of your body and goes into your body, delivers its genetic information, causes you to have a cold. Back in my microbiology days, I thought, wouldn't it be great to engineer one of those viruses and take advantage of its ability to get into the human body, but now deliver genetic information that can be useful? And so here's a schematic of what this actually looks like. And so with lots of work, I and others put together a scheme to develop a new virus that's based, this is an adenovirus, it's a cold-causing virus, take apart its elements, its DNA elements, and replace them to make it safer as well carry genetic cargo as a medicine. And this is we don't have many of these, but this is a eureka moment for me because this is the first time I actually produced this virus that theoretically we thought we could make to scale. So in that little white band there is 1 followed by 14 zeros of virus. This is an engineered manufactured virus. So at that day, I knew that this could be manufactured to scale, and that actually represents 10,000 doses, okay, in that little tube there. And so now, based upon that, we have started using this virus as a immunotherapy to treat cancer. So we can deliver genetic information to patients that are end-stage cancer patients to stimulate their immune systems to fight their cancers. This is called precision medicine, if you will. And what I pulled up here, this is actually a slide that was about a month old. It's actually out of date. Now there's about 28 FDA-approved clinical trials using this technology to treat colon cancer, breast cancer, pancreatic cancer, gastric cancer, all of these cancers using this technology alongside a number of other immunotherapies. This is very novel to bring multiple experimental things together at once to treat some of these most difficult of cancers that have yet to yield to clinical medicine. And so that's how something moves from an idea into clinical trials. Now we'll see if this can actually improve lifespan, improve the outcomes for these patients, and hopefully we get to another drug that can be prescribed by patients, physicians worldwide. Finally, a third example. This is a thing we study in my laboratory, an autoimmune disease called Ankylosing Spondylitis. This is, again, a debilitating autoimmune disease. That means your immune system is attacking your own body in various areas, in this case, the spine, to cause a crippling fusion of the bones. And this affects teenagers when their first presentation. It can get very progressive to the point where you're debilitated, can't move, maybe wheelchair bound. Now, I'm a geneticist, and we know that there was a gene associated with many families that had this condition. It's called ERAP1, and I'm not going to go into the details of it. All we knew is that this gene was more present in individuals that had this condition than those who did not have the condition. But that's where it ended. Really, we had this association in the literature. No one knew what it meant. So in my laboratory, we actually took that gene, mutated it in mice, taking advantage of some of the technology we have on campus, new imaging. That's a mouse on the left there, and we did a micro CT scan. This is a CT analysis that maybe many of you had, and applied it to these mice and found that these mice actually did develop the abnormal fusions in their spine just like the human patients do. So now we have a model that we can investigate much more easily in the laboratory, and it turns out that we found that these mice have a deficiency in a type of immune cell that actually puts the brakes on the immune system. So your immune system sort of like running a car and hitting the gas, the brake, it's at the same time, it's modulated to regulate how intense the immune response is. And in these patients, they have a deficiency in the cells that put the brakes on the immune system. We didn't know that before. And so now we will be moving forward with that knowledge to potentially increase the number of these cells in these patients to treat their autoimmune disease. Now, the beauty of this is that ERAP1 gene that I talked about is also associated with other conditions that you may have heard of, like multiple sclerosis, non-insulin dependent diabetes mellitus, psoriatic arthritis. So this knowledge will now, and this insight will now be targeted to all these diseases, not just ankylosing spondylitis. So with that, I'll stop, and I thank you for your attention, and we'll take any questions.
All right. Who has questions? How many cases of Pompe disease? How many?
It's about 10,000 worldwide. 5 to 10,000, at least in the United States and Europe.
Yeah.
It's about 1 in 30,000. There's probably more, though, because many of these children were never identified. They may have been a SIDS child, that died of a respiratory failure. And if you weren't looking for it, you wouldn't know. Now that we have a therapy, though, physicians are very well aware of what to look for in these children. And the thing is, it doesn't just affect infants. It can affect, I think about it. Tomorrow I could wake up and I can't go up the stairs like I used to, and it's the first signs that I actually have Pompe disease. I've got patients right now. I treated one yesterday. She's 80 years old with Pompe. She presented when she was in her 50s. So there's a spectrum, and that's the other thing about these diseases. Sometimes the patients don't read the books on how they're supposed to present. We have to learn as physicians to listen to our patients and identify them.
I know that Michigan takes that blood test, right?
Yeah.
Is that that drop of blood is what?
Yeah. Yeah, that simple spot of blood, we take that, actually take a portion of that spot of blood, run it through a laboratory test, and we can identify the enzyme that's missing, alongside about 80 other conditions, many of which are genetic, that we screen for right now. In fact, the state of Michigan is probably cutting edge on newborn screening. It's been out in front for many years, so it's been fun to be a part of that. Anything else?
Anything? Board?
Okay.
Thank you.
Okay, well, thank you for your time.
Appreciate it.